Hey everyone, so we are in our final class of the week. This is our first week sort of ticked off. We did it. I would love to hear about how things went for you. If you had any trouble finding things, if you had any trouble following stuff, please let me know. Again, I'd like to be kind of in a state of continuous improvement for this course, especially in the early stages of adapting to online learning. So for this weekend, what I'm going to task you with is a whole bunch of reading and a bunch of thinking and good stuff. So I'm going to give you another excerpt from that book, Braiding Sweetgrass, that we read the first day. Look for the passage called Epiphany in the Beans. This is uploaded to your week 1 modules tab. You can find it there, and it will also be linked into your Thursday checklist assignment page. So you'll be able to find it that way also. Should you have any trouble, let me know, of course. You also have some reading from the OpenStax textbook. So this is going to be a different OpenStax than the one that we have been using. And this is because the topic that we're getting into just happens to not be covered in the one that we've been using. So that link is provided in your checklist also. I've also given you a short video on this really interesting theory in evolution called the endosymbiosis theory. So folks who took my class last term, I think we may have spoken about this a little bit, but I don't want to, ruin the surprise for anybody for whom this is totally unfamiliar. The next thing that you're going to do, and the last thing that I'm going to have you do, is start your video project. So you're getting a video project, and this is going to be due in about 2 weeks. The details are uploaded in a separate assignment, which you can also find in your Thursday checklist assignment page. And then I will also be uploading a short video just sort of talking through the expectations also, as well as demonstrating a couple of the tools that I recommend that you use for this. So look for that. I may upload it this weekend a little bit early, or at the very least it will be available Monday. So you'll be able to take a look then. But start checking out that assignment and see what that looks like. Check out some of the resources that I've recommended for you, and let me know if you have any questions in advance of that video. And as a reminder too, I do hold conference periods on Monday, Tuesday, and Wednesday right now. So that's a really great time to just sort of quickly check in and get some, I guess, live help. And I'd love to see you also. So that's always a thing. So let's look at our stuff for today. So the last time we spoke, we were sort of going through the process of photosynthesis, and we tapped out a little bit early because it's quite dense, and I wanted to give you a little space to process. We're kind of talking about like straight up molecular biology right now. If the unit on cellular respiration was something that worked pretty well for your brain, this likely does too. It's a very similar mechanism. So where we left off was we were sort of describing this process of chemiosmosis. And so what had happened was light comes into photosystem 2 here and excites the electrons in the chlorophyll molecules. And then that excitation, that excess energy, then becomes transferred from chlorophyll molecule to chlorophyll molecule to chlorophyll molecule, all the way down, transferred by electrons until we reach the center of the photosystem. And I'm just going to quickly switch to the laser pointer because my cursor keeps vanishing. I hope that's maybe easier to see. So we wind up in the center of the photosystem here, and then those excited electrons are going to be sort of launched up to the electron acceptor here. Once they've reached that electron acceptor, they're then going to begin, I can't talk right now, their passage through the electron transport chain, which is this piece of membrane here, which includes 2 electron acceptors. There are actually a little bit more than 2, but in our diagram, there are 2 electron acceptors, as well as a transmembrane protein whose job it is, again, to pull protons from the stroma, which is up here, to the thylakoid lumen. So lumen is sort of a generic term in biology and physiology specifically. Lumen is what we would use to describe the interior of an organ, or specifically the interior of something that is membrane bound. And so we would say, like, the thylakoid interior, thylakoid space is a synonym to thylakoid lumen. And so we are using the energy generated from the passage of electrons across the electron transport chain, moving towards photosystem 1 here, to pull protons from the stroma of the chloroplast to the thylakoid lumen. And so what's happening here is, as we begin to build up a concentration of protons, as we begin to build up that, we can almost think of it as osmotic pressure, really, if we think back to learning about osmosis and concentration gradients. What we're really doing here is we're creating a concentration gradient. And so all of these protons get pulled down here. They're building up, building up, building up. And when they do, remember, this transmembrane protein that they came in through is a one-way street. It doesn't allow protons to just leave the way they came. So now we've got this excess of protons here, very highly energetic. They don't want to all be close to each other. They want to be able to diffuse out to an area of low concentration. And the area of low concentration, now that they are highly concentrated in the lumen, the area of low concentration is going to be in the stroma. And so the only way out for these protons, the only direction that they can diffuse, is going to be through this molecule here, which we call ATP synthase. And so more on that in just a moment. So the passage of these protons is going to, the passage of the protons through ATP synthase is going to create a chemical reaction that is going to join a phosphate ion to the molecule ADP. So we said that ATP's full name is adenosine triphosphate. So adenosine kind of sounds like adenine, which is one of the nucleic acids that we learned about last term. And chemically, they're pretty similar. So adenosine, and then joined to it, we have 3 phosphate molecules. So tri meaning 3, so we say that is adenosine triphosphate, adenosine plus 3 phosphate molecules. And so its cousin, ADP, is actually its precursor. So ADP is going to be adenosine diphosphate. So adenosine molecule joined to di meaning 2, so 2 phosphate groups. And so the job of ATP synthase, remember, if we think about breaking these words down, anytime we see the suffix ase, A-S-E, we recognize that as an enzyme molecule. And so that tells us something about its structure and function. And so we have the ase, synth sounds like synthesis, right? And then ATP, so a molecule that synthesizes, or an enzyme that synthesizes ATP. And so as these protons are passing through ATP synthase, they turn the turnstile at the top of the molecule, which creates the energy required to join that 3rd phosphate group onto ADP, creating an ATP molecule, so adding that 3rd phosphate group. And so the electrons that sort of made their way over. We talked about the protons accumulating in the lumen, but we didn't, really address the rest of the journey of the electrons. So remember, they went up to the electron acceptor, they passed through the electron transport chain, lending their energy to this transmembrane protein to pull in protons. And so now these electrons that have lost some energy, and that energy again was picked up by that transmembrane protein, those now de-energized electrons are going to get picked up by photosystem 1 here. And so photosystem 1 is going to use, again, light energy, not pictured here, but light energy hitting these chloroplasts, exciting these electrons, and then picking up the de-energized electrons from the electron transport chain, re-energizing them, shooting them up to the electron acceptor here, where they can then move over to the molecule NADP+. And so NADP+ then undergoes this process that we call reduction. So I think we talked about a little bit in the last video, reduction in chemistry actually refers to a reduction in charge rather than a reduction in anything else, in quantity or in something else. So reduction in chemistry refers to reduction in charge. And so if we have a molecule NADP+, well, that plus indicates that it has a positive charge overall. That molecule has a positive charge overall. And so by adding an electron to it, which has a negative charge, we are then neutralizing that positive charge of the molecule, leading to an overall reduction of charge. And the acronym I gave you to remember this is OIL-RIG. So we haven't really talked about oxidation. I think we may as we go throughout the term, but OIL-RIG stands for oxidation is loss, reduction is gain. And so the loss and gain refer to loss and gain of electrons. And so in this case, we're focusing on the RIG part of that. So we would say that reduction is gain. So we are reducing NADP+ through the gaining of an electron, which yields NADPH. Now, this molecule is going to be really important for the Calvin cycle, as we'll see shortly. And so as the electrons are lost from photosystem 1, remember they're getting lost to reduce NADP+, creating NADPH, they are continuously replaced by the electrons that are being transported across the electron chain. So this is cyclical. As we lose electrons to the formation of NADPH, we are continuously gaining replacement electrons from that electron transport chain. And so the cycle has to include other components, right? Because there isn't just an unlimited supply of electrons within the within the chlorophyll molecules, right? They don't just have an unlimited amount of electrons that they can continuously just be sending across the electron transport chain. And so one thing we know about plants is that they need water. They have to have water. If we don't water one of our plants, it's going to wither up and die. And the reason for that is because water is actually directly responsible for fueling the production of ATP and NADPH, therefore is directly responsible for creating the energy required to sustain a living plant. And so here's why. What happens is water is taken up by the roots of the plant. This mechanism can vary depending on the species of plant. We'll talk a little bit about that later. But so water is taken up and it eventually makes its way to the thylakoid lumen. So remember, the thylakoid lumen is inside the thylakoids, which are stacked as grana, which we find on the inside of the chloroplast. And so the chloroplast is then inside the cell. So water has to essentially diffuse into the plant cell, make its way into the chloroplast. And so these molecules of H2O, this water, is making its way to the thylakoid lumen, where it sort of, I guess I like to think of it as like it like slaps into the energized electron here of photosystem 2. So the light again is coming through, it's energizing electrons all the way down, eventually making its way here. And then the H2O molecule, using energy from light photons as well as the energy from the electron here that it's sort of slapped into, that H2O molecule is going to bust apart. And when it busts apart, it's going to break into its constituents. So the electron from the water molecule is actually going to go into photosystem 2 and eventually be sent down the electron transport chain, where it can power the pulling of protons from the stroma to the thylakoid lumen. Now, that's not the only thing that's in water. We also have 2 hydrogen atoms. And so we'd remember from chemistry that H2O, the subscript 2, indicates that there are 2 hydrogen atoms as part of this molecule. And so those 2 hydrogen atoms, those 2 protons, when that molecule is busted apart, those 2 protons are going to remain in the thylakoid lumen. And so their accumulation is part of, or their presence is part of that accumulation of protons that will eventually be sent through ATP synthase to power the production of ATP. And so that accounts for 2 of the ingredients, but the third one, maybe the most important, relevant to humans, is going to be this oxygen atom. And so what they're saying here, they're notating this as half of O2, because if we look at the molecule of water, we see that there are 2 H atoms, but there is only 1 O atom. Now, oxygen actually likes to exist in a pair. It doesn't generally exist as just like a single oxygen atom. It's usually going to want to be 2 oxygen atoms. So when we notate this, often we are showing that we're often writing oxygen as O2 rather than just O. And so this is why they have notated half O2 rather than just O. And so we have, again, the electron from the water molecule being sent into photosystem 2, the protons accumulating within the lumen, and then the oxygen, well, that gets released as a byproduct. So oxygen, we hear about plants producing oxygen for us all the time. That oxygen is actually a waste product of the light-dependent reactions of photosynthesis. And without that, we probably wouldn't have an atmosphere as we know it, and therefore we probably would not have the diversity of life as we know it on Earth. This process is what we call photolysis of water. And so photo, again, meaning light, lysis meaning to break. And so this is the breaking of water using light energy. So we spent a lot of time talking about the light reactions. I hope that is feeling maybe semi-understood. If not, come see me and let's talk about it. Totally fine. There's lots of video resources out there if you would rather use something like that. But I would love to help you in your understanding of this also. So that is just one half of the cycle. We also have the light-independent reactions, also called the Calvin cycle, which is sort of the next phase of photosynthesis. And so I guess if you like start looking around on the internet, you'll hear a lot of people complaining about the Calvin cycle, saying that it's like really hard, it's really complicated. Maybe you'll think that about it. It looks certainly very complicated this way, but it's actually fairly simple if we break it down. So the first thing that's going to happen, well, I guess the first thing we need to acknowledge is that it's a cycle. So things like beginning and end like don't really mean anything. So we're sort of picking an arbitrary starting point. Depending where you look, you may see the starting point defined differently. We are going to define our like starting point as the entry of CO2 molecules to the Calvin cycle. And so we talked about these structures, stomata, these pores in the leaves of cells, and we said that these are the structures that are necessary and responsible for gas exchange within a plant. And so these stomata are continuously exporting oxygen, that is the waste product of the light-dependent reactions, and then intaking CO2, that is necessary for the beginning of the light-independent reactions. You may also hear these called the dark reactions, which I always thought was kind of a cool name. So CO2 anyway is going to enter this Calvin cycle. It's going to enter these dark reactions, and when it does, it's going to react with an enzyme called Rubisco, which is sort of written like this, which always reminds me of that SpongeBob meme, like the mocking one. But anyway, so CO2 is going to react with Rubisco, and Rubisco is an enzyme that is used in this reaction. Now, what we need to know about enzymes for right now is that enzymes are molecules that facilitate chemical reactions that do not get consumed or degraded in the process of that chemical reaction. So we would say that an enzyme, while it is used to initiate a chemical reaction, is not, is conserved in the process of that chemical reaction. This is something you may have seen in chemistry, you may have seen it in earlier terms of bio, but just in case not, just making sure everybody's on the same page. So Rubisco and RuBP are going to be the two enzymes that we really need. And so the first stage of the Calvin cycle is what we call fixation. You may see it as carbon fixation, carboxylation. We're going to refer to it as just fixation. And this is the catalyzing of a reaction between RuBP and 3 molecules of carbon dioxide that have entered the cycle using Rubisco. So Rubisco is kind of like the matchmaker for RuBP and carbon dioxide. And so through this reaction, a 6-carbon molecule is formed, which we quickly split up into 2 3-carbon molecules that we are going to call 3-PGA. And so 3-PGA is formed. And the next stage of this cycle is what we're going to call reduction. And so again, reduction in chemistry means a reduction in charge. And so what's going to happen here is PGA, those 3-carbon molecules, are going to react with both the energy from ATP and electrons donated from NADPH, which yields ADP and NADP plus respectively. Those are going to, the contributions of those 2 molecules are going to convert 3-PGA into another molecule that we call G3P. Now, G3P is also a 3-carbon molecule. It is slightly different. We won't get too deep into the biochemistry of that, but it is we can think of it as another version of a 3-carbon molecule that's going to go through this process. Now, once we have created a DP and NADP plus, those are actually going to return to the thylakoid lumen. So those are going to return back to where they came from, so that they can be recycled as part of that, as part of those light-dependent reactions. And so the G3P molecule is gonna have some pretty important roles here. So we can kind of think of this entire process, like leading up to here, as being for the purpose of creating G3P. And so one of those G3P molecules is going to just get kicked out of the Calvin cycle. And so when it does, it's going to be sent off to somewhere else that we're not going to get into, but it's going to be sent off to get assembled into a glucose molecule. Now, glucose is made of 6 carbons. It is a sugar, so it's a carbohydrate molecule, and it's for the purpose of storing energy. That glucose molecule will eventually be sent to the mitochondria to undergo the process of cellular respiration, which you learned in the fall. And so when it undergoes that process, it's going to produce a lot of ATP for the cell. And so that's what's going to happen with one of these per turn of the Calvin cycle. So every time this cycle goes, we produce one G3P that's going to get kicked out, eventually become glucose. All of the other G3P molecules that were produced through this first half, through the carbon fixation and the reduction that occurred prior, the other molecules of G3P are going to be recycled, and they are going to be involved with this process that we call regeneration. Specifically, this is the regeneration of Ruby P. And so Ruby P is going to, or that process of reforming Ruby P is going to require the additional energy input from more ATP molecules, which is going to yield additional ADP, which can be sent back to the light-dependent reactions to become reformed into ATP, so having 3 phosphates. And the reformed Ruby P can then just re-enter the cycle, which can continue to react with CO2 and continue this entire process, creating more G3P, which will go on to glucose production each time that it goes. And so that's it. I mean, that's the process of photosynthesis. You know, again, photosynthesis includes both the light dependent reactions as well as the dark reactions, the Calvin cycle. And so all of this is what makes plants really special. So plants are the only ones that can do this, mostly. Photosynthesizers have been really important historically for the evolution of life as we know it. And so looking at some of those early photosynthesizers, the oldest one that's ever been documented is an organism that we call cyanobacteria, and they are about 3.5 billion years old. That's super, super old. And so I think the really cool thing about them is that we still see them around today. In fact, if you've ever seen a lake or a pond that looks like this, you have seen cyanobacteria. They are generally fine. I mean, for people, they're generally non-pathogenic, so they typically cannot make you sick. But occasionally we do have cyanobacteria that will produce toxins as, you know, a byproduct of their metabolism. And so those toxins can be harmful to humans, or they can be harmful to other life living in those ponds. So I mean, they're having a great time when you see a pond that looks like this, but generally things that live there are probably not having an awesome time. So you won't get sick from these, like, per se, like you won't get an infection from a cyanobacteria because your body is not well suited for their survival, but it's almost like a side effect of their existence, right? Like they produce these compounds which can make us sick, that can be toxic to us. But these are actually, because they were the first photosynthesizers, these are going to be the first organism that is actively producing oxygen. And I almost can't stress enough how just awesome and important that is. Without their existence, without them photosynthesizing, harvesting energy from the sun, we would not have oxygen as part of our atmosphere, and therefore we could not exist. Human life as we know it, any land animal life as we know it, as it has currently evolved, could not exist this way. It's impossible to know what could have happened, but I do think it's really incredible to sort of acknowledge and appreciate the series of events that led to our existence that could not have happened any other way that we know of, at least. And so what's, I think, really cool about cyanobacteria, and what I'm having you guys watch a short video on, is that cyanobacteria are actually thought to be the evolutionary grandfather of chloroplasts. And so because these were the first organisms to photosynthesize, I mean, they're little, they resemble they are bacteria. They sort of get mistaken for algae very often. They get, mistakenly called algae. They're not quite, they're prokaryotes, where an algae would be a eukaryotic, single-celled plant. And so, but if we look at the structure of cyanobacteria, they actually really closely resemble chloroplasts. And so it's thought that their existence and their evolution sort of converged with early plant cells, and they formed this sort of symbiotic relationship that led to eukaryotic cells being able to photosynthesize. So there's a whole video about that. It's called endosymbiotic theory. It's very, very cool. Definitely check that out. But so following our cyanobacteria, we didn't stay in the water forever. Photosynthesizers didn't stay in the water forever. They eventually had to adapt to life on land and become organisms that were capable of adapting to life on land. And they did this to occupy an ecological niche that was not yet occupied. And so the first adaptation that led to this, the first trait that they evolved that was able to support this, was the evolution of the cuticle. So we talked about the cuticle a little bit last time. The cuticle, again, is this really waxy covering that helps the plant by maintaining homeostasis through preventing water loss through the leaves and through the outer parts of the plant. And so when they evolved this cuticle, now they were able to sort of retain water a little bit better. So the plants that most resemble those early land plants that we can still see today are going to be like your mosses. So if you see a moss just growing out in the world, that very closely resembles the first mosses that ever were. There's also another plant called liverworts. They're actually like a category of plant. You may or may not have seen them. You'll sometimes catch them in like a deep dark forest. They're usually like quite low. They don't grow very tall, but liverworts are quite ancient also. And these guys evolved about 470 million years ago, so quite a lot of time after cyanobacteria, but still quite ancient and old. And so when these guys came out of the water, and they evolve their cuticle and they're able to survive long enough to reproduce and become reproductively successful, they're just photosynthesizing all the time. And so their existence contributed to the oxygenation of the atmosphere in the most significant way that we know of. The interesting thing about them is that they actually lacked a vascular system, and still do lack a vascular system. And so they actually have to rely on things like rainwater for reproduction. The reproductive cycles of land plants is not something that we're going to talk a lot about, or at least we're not going to talk a lot about the reproductive cycles of things like mosses and liverworts, things that lack a vascular system. But it is something that is interesting to know, is that they actually require water for their reproductive system. And so that's where I think we'll leave off for today. So please, come see me with any questions that you have. This is kind of a lot of information, and then we sort of switched gears here right at the end. So please come see me and let me know what questions you have, what I can help with. Let me know how your first week went, and if there's anything else I can do, would love to hear that too. But hopefully I'll talk to you soon. Enjoy your weekend, get lots of rest, stay organized, wash your hands. I will talk to you soon.